Welcome to Lakshman Biology. Now we will start the new chapter, that is chapter number 5 in second PUC Biology, that is Principles of Inheritance and Variation. Principles of Inheritance and Variation. These Principles of Inheritance and Variation chapter also called as Genetics. Genetics are Principles of Inheritance and Variation. Okay, first we have to see the few scientists which did work on the principles of inheritance and variation, or who did work on the genetics. Okay, that is number 1, G.J. Mendel. Mendel is the scientist, he called as father of genetics. Mendel is the father. Which father? Genetics father. Father of genetics is Mendel, father of genetics. Already we know that the genetics are the chapter number 5, that is Principles of Inheritance, also called as genetics. Who is the father of this genetics? Father of genetics is Mendel. He worked on the pea plant and he explained how the characters are transferred from one generation to another generation by 3 laws. Hence, the Mendel is called as father of genetics. Then Bateson. The Bateson called as father of modern genetics. Father of modern genetics. And the term genetics also given by the Bateson. He also given the term genetics. That is about Bateson. Next, T.H. Morgan. T.H. Morgan is called as father of experimental genetics. Father of experimental genetics. Experimental genetics. Who is the father of experimental genetics? That is T.H. Morgan. T.H. Morgan worked on the Drosophila or fruit fly, and he proposed the term linkage, and he proposed the sex linkage, crisscross inheritance, etc. Hence, T.H. Morgan called as father of experimental genetics. He did experiments on the Drosophila. Next, A. Garrard. A. Garrard is the scientist, he called as a father of human genetics. Father of human genetics. And he identified first metabolic genetic disorder, that is alkaptonuria. Alkaptonuria. Alkaptonuria is one type of genetic disorder. This alkaptonuria is identified by the A. Garrard. He is the first scientist who identified metabolic genetic disorder. He identified the alkaptonuria. What is alkaptonuria? Alkaptonuria is genetic disorder in which there is an absence of one enzyme that will be degenerate, that will be break the homogenistic acid. Okay, there is one enzyme which will cleave the homogenistic acid. That enzyme which will digest homogenistic acid is absent. Because of that, we can see the accumulation of homogenistic acid throughout the body. Why? Because of absence of enzyme which will cleave the homogenistic acid. Okay, why that is absent? Because of absence of the particular gene which produce enzyme for cleavage of homogenistic acid is mutated. Because of that, there is a accumulation of homogenistic acid in the body, leads to one disease, that is called alkaptonuria. Once again, alkaptonuria is one type of metabolic genetic disorder in which is called because of absence of one enzyme which will break the homogenistic acid. Okay, next we should discuss about the terminology of genetics. For better understanding of the chapter genetics, we should know about the few terminology which is related to the genetics. First, we have to discuss about the terminology which is related to the genetics. After that, we will discuss about the remaining parts which are present in the genetics. First, terminology. In first terminology, okay, as it is, we will see about the chapter name, that is genetics. What is genetics? First we should that is the first word that we have to know, genetics. What is the genetics? genetics is branch of biology which deals with genetics is branch of biology which deals with the study of inheritance and variation, which deals with the study of inheritance and variation. Already the chapter name only inheritance and variation. Genetics means study of inheritance and variation. That is the genetics. It is the branch of biology which deals with the study of inheritance and variation. What is inheritance here? What is inheritance? Inheritance means transfer of characters. Transfer of characters. From where to where? Transfer of characters from parents to offsprings. From parents to offsprings is called inheritance. Okay, we can say heredity also. Heredity meaning transfer of characters, and inheritance also the transfer of characters from one generation to another generation. Which is basic unit which is used for transfer of characters from one generation to another generation? That is gene. Gene is small stretch of DNA, or it is the basic unit of inheritance. Because of presence of gene on the chromosomes, because of presence of genes on the chromosomes, the characters will transfer from one generation to another generation. That is about the inheritance. Inheritance means transfer of characters from one generation to another generation. Then variation. What is the variation? Variation means difference. Difference in between parents and offsprings. Parents and offsprings. Offsprings means children's. And difference between offsprings of same parents also. Offsprings of same parents. This is also variation. If you see, because of sexual reproduction, the sexually reproducing organisms show the variations. What is that variation? There is a difference in height, difference in size, difference in shape. Those are called variations. Parents and offsprings, they may show the little differences in few things. That is called variation. If the offsprings are children which born from the same parents also different. If 5 children are there, all the 5 children are not the similar. There is a few differences are there in size, shape, color of that children. That is also called variation. Here, that variations are mainly divided into 2 types, that are somatic variations. Variations which occurs in somatic cells other than germ cells. Somatic cells means the cells which are not involved in the formation of gametes. Those are called somatic cells. If variations occurs in somatic cells, that is called somatic variation. Then if variations occurs in germ cells means the cells which are involved in the formation of gametes. If variation occurs in germ cells, that is called germinal variations. Germinal variations. Germinal variations means variations occurs in the germ cells. These germinal variations again divided into 2 types, that are continuous variation and discontinuous variation. Continuous variations occurs continuously. The continuous variation is because of crossing over during the formation of gametes, or because of recombination. Because of crossing over and recombination, we can see the continuous variation. What is the recombination? There is a exchange of genetic material in between the non-homologous chromosomes is called recombination. There we can see the variations in germ cells. Hence, that is continuous variation. Next, discontinuous variation. In few types of organisms, in few conditions, you can see mutations or leads to variation in germ cells. That type of variations are called discontinuous variations. The variations which are occurs in somatic cells or somatic variations will not transfer from one generation to another generation. But if you see the germinal variations can transfer from one generation to another generation. Why? Because of gametes only we can see the formation of new organism. If the gamete mother cells or if the cells which are involved in the formation of gametes get mutated or get changed, then that will transfer into the next generation also. Hence germinal variations are inheritable and somatic variations are non-inheritable. Okay, because if you see in our surroundings there is a crossing by the selected animals, that is called artificial selection. Because of artificial selection means in artificial variations we can see because of artificial selection also. Example, if you see different types of dogs in our surroundings, different variety of dogs are there. How that dogs are formed? Because of artificial selection. Okay, we have to select the male and we have to select the female. Then if you cross that desired male with desired female, you can get a superior offspring. That is called artificial selection. Example for animal which is formed by the artificial selection is a Sahiwal cow. Sahiwal cow is the cow which has better characters. Okay, the variation in offspring we can see because of artificial selection also. Because of artificial selection also we can see variations. Okay, example for that is there is one cow, that is Sahiwal cow, which is present in the Punjab, which is formed in Punjab. How it formed? By the domestication and artificial selection. By artificial selection and domestication we can see the formation of better varieties species. Example for that is Sahiwal cow, which is formed by the domestication and artificial selection. Based on this we can understood that our ancestors also know about variation. How they know? Little bit they know. How they know? They know that if they cross better variety animals, then there is a better variation which has a better characters. They can see, we can get, they know that we can get in the offsprings. That is about the variation. After the variation we should discuss about the chromosome. Chromosomes are thread-like structures. Thread-like structures which are made with proteins plus DNA, which are made with the proteins plus DNA. How many chromosomes are present in human beings? Human beings contain 23 pairs of chromosomes. 23 pairs. 23 pairs of chromosomes are present in human beings. Okay, 20. Why we are saying 23 pairs? Because chromosome number 1 is similar, chromosome number 2 is similar to each other. Chromosome number 3 pair. Okay, each chromosome occur in a pair. This is chromosome number 1. It is occurs in a pair. Hence, we have to say 23 pairs of chromosomes. These similar chromosomes are called homologous chromosomes. Homologous chromosomes. What is the homologous chromosomes? The chromosomes which has same genes at same loci. Loci means place where genes are present is called loci. If same genes are present on same place of two chromosomes, the two chromosomes are combinedly called as homologous chromosomes. Okay, chromosome 23 homologous chromosomes, 23 pairs of homologous chromosomes are present in human body. Last pair only XX or XY, that may be different, but remaining all are occurs in a pair. Each pair is called homologous chromosomes. That is about the chromosome. Chromosomes are again divided into two types. If you see in human beings, 22 pairs of 22 pairs of chromosomes are called autosomes, or body chromosomes, or somatic chromosomes, and remaining one pair of chromosomes are called allosomes or sex chromosomes. XX or XY are sex chromosomes. Remaining 22 pairs of chromosomes are autosomes. Another name for autosomes is somatic chromosomes. Another name for allosomes is sex chromosomes. Human beings have 22 pairs of allosomes and one pair of 22 pairs of autosomes and one pair of allosomes. That is about the chromosome. Next, factor or gene. Okay, what is the factor or gene? It is basic unit of inheritance. Basic unit of inheritance. Because of factor or gene only, we can see the transfer of every character from one generation to another generation. Hence, we can say that is basic unit of inheritance. Apart from that, gene we can also call as small stretch of DNA. Small stretch of DNA. If you see eukaryotes like human beings, they contain approximately 2.2 meters DNA. On the DNA, we can see that small segments are called as genes. Those are helped for the transfer of characters from one generation to another generation. That is about the factor or gene. Mendel called the name factor. Okay, Mendel don't know about name gene. He called them as factors. After that, another members called as a gene. The term gene is given by Johansen. Johansen given the term gene, and term factor is given by the Mendel, and the structure of gene is identified by the Benger. If you see the structure of gene, each gene has 3 parts, that is a recon, muton and structural gene. The structural gene also called as a cistron. If you see the prokaryotes contain lot of cistrons. Hence, prokaryotic gene also called as polycistronic gene. Each cistron will produce one protein. Eukaryotes contain only one cistron, and hence eukaryotic gene is called a monocistronic gene. That is about gene or factor. Once again, the term factor given by the Mendel. Next, gene is given by the Johansen. Structure of gene is proposed by the Benger. Gene is basic unit of inheritance, which is the small stretch of DNA. Next, we have to see about alleles. What are alleles? Slightly different forms of gene, or simply you can say that forms of gene. See, each gene occurs in a pair. I told that 2 homologous chromosomes are present in human body. See, this is homologous chromosome. Same type of genes present on each chromosome. Okay, each one is called allele. Slightly different forms of gene. Capital T, small t are 2 forms of one gene. Capital T is dominant allele, small t is a recessive allele. That is the alleles. Forms of gene. Each gene or factor occurs in a pairs. Okay, if that pairs are different, one is dominant, another one is a recessive. Those are called alleles. See here, next, dominant allele. Already we know that genes occurs in a pair. One is dominant allele, another one is a recessive allele. What is dominant allele? Dominant allele is the allele which express its phenotypic character in the homozygous condition and in a heterozygous condition also. If capital T, small t, those are 2 alleles which will responsible for the height of plant. Here, capital T will give tall plant only, and capital T, small t also give tall plant only. This small t is recessive allele. The capital T suppress the recessive allele phenotypic character, and it will be exhibit in the presence of another recessive allele also. Okay, that is about dominant allele. Dominant allele always expressed with a capital letter. Once again, dominant allele is the allele which express its character in both homozygous condition. If both are capital T's also, it will exhibit tall plant only. If one capital T, one small t also in the presence of another recessive allele also, we can see the formation of a tall plant. That is about dominant allele. Recessive allele is the allele which express only in homozygous condition. If both small t's are there, then only dwarf plant is formed. Okay, if one capital T, one small t is there, which leads to formation of tall plant only. Why? Because one dominant allele is there. This dominant allele suppress the recessive allele phenotypic character. Both capital T's also leads to give tall plant, for formation of tall plant. That is about recessive allele. Recessive allele always expressed with the small t. Next, homozygous alleles. If two alleles are same, that is called homozygous alleles. See, this is one homozygous condition and this one is another homozygous condition. This homozygous again divided into two types. Homozygous dominant means if both dominant alleles are there, that is called homozygous dominant. If both small alleles or recessive alleles are there, that is called homozygous recessive. Homozygous again divided into two types. Homozygous dominant means both dominant alleles are there. Next, homozygous recessive means both recessive alleles are present. That is about the alleles: dominant allele, recessive allele and homozygous. Next, heterozygous. Heterozygous alleles or heterozygous genotype means one dominant allele, one recessive allele. It may be capital T, capital W, small w, or it may be capital T, small t, or it may be capital R, small r. One dominant allele along with one recessive allele. If both are present, that is called a heterozygous allele. This condition is called heterozygous dominant. Why? Because which will give phenotypic character dominant only. Hence, it is called a heterozygous dominant. That is about heterozygous allele. Next, phenotype. Phenotype means morphological appearance. Morphological appearance of gene. Morphological appearance of gene is called a phenotype. Example, capital T is one genotype which will give phenotype that is tall. Small t is one genotype which will give dwarf plant, okay, which leads to formation of dwarf plant. Next, capital T, small t, that is heterozygous dominant. It is one genotype which will give the tall plant. Morphological appearance, we can see color, size, shape, that all are called as morphological appearances. That is called phenotype. Genotype is responsible for the phenotype. Genotype only determine which type of phenotype has to form. See, if in one plant, inside this plant both capital T's are there, it become into tall. Why? Because which contain both capital T's. Both small t's are there, it will become into dwarf. These capital T, small t's and capital T's are genotypes. That genotypes, based on type of genotype only, phenotype is determined. That is about the phenotype. Next, genotype. Genotype, genetic constitution. Genetic constitution or genetic makeup. Genetic makeup of an organism is called a genotype. It may be homozygous dominant, or it may be heterozygous dominant, or it may be homozygous recessive, which is responsible for the formation of a genotype. Next, hybrid. Offspring. Hybrid means offspring. Offspring which is formed by the crossing of two parents. Which is formed by the crossing of two parents. Children which formed by the crossing of two parents is called a hybrid. Next, hybridization. Hybridization means cross between two parents. Cross between two parents which differ, which will differentiate in one or more characters. Okay, cross between tall plant with the dwarf plant, cross between round seed plant with the wrinkled seed plant. Okay, cross between two parents which are differentiate in single character or more character. Cross between tall plant with the dwarf plant, that is a single character. Okay, that is called hybridization. See, once again, hybridization means cross between two parents which are differ in one character or more than one character is called a hybridization. If we cross the parents which are differ in one character only, that is called monohybridization or monohybrid cross. If you cross the animals, the animals or organisms or plants which are differ in two characters are called dihybrid cross. or dihybridization. Next, true breed, the breed which formed by the continuous selfing. Selfing means cross between offsprings of same parents. Cross between offsprings of same parents are called selfing. Because of continuous selfing, we can get homozygous condition. Okay, capital T or small t like that. That is about the true breed. True breed is the breed which contain homozygous condition. Because of that, we can get the true breed which contain homozygous condition. That is about the terminology of genetics. Once again, okay, Mendel is the father of genetics. Bateson is the father of modern genetics. Morgan, father of experimental genetics. Gerard, father of human genetics. Next, if you see terminology, genetics, meaning of genetics, which deals with the study of inheritance and variation. Inheritance means transfer of characters from parents to offspring. Variation means difference in between parents and offsprings, in between offsprings also. Variations are two types, somatic variations and germinal variations. Somatic variations are not inheritable. Germinal variations are inheritable, means they can transfer from one generation to another generation. Next, chromosome, thread-like structures which are present in nucleus, which made with DNA plus proteins. Factor or gene, it is the basic unit of inheritance. Alleles, alleles means different forms of genes. Next, dominant allele means which expresses its phenotypic character in presence of alternate allele also. Recessive allele, which always express phenotypic character only in homozygous condition. Next, homozygous alleles, if both alleles are similar type, that are called homozygous alleles. These are again divided into two types, homozygous dominant and homozygous recessive. Heterozygous alleles, if both alleles are different, that are called heterozygous alleles. Next, phenotype, it is morphological appearance of a gene. Genotype, genetic makeup of an organism. Hybrid, offspring which is formed by the crossing of two parents. Hybridization, cross between two parents which are different one or more than one characters. If we cross the parents which are different single character, that is called monohybridization. If you cross the two parents which are different two characters, that is called dihybridization. True breed, the breed which has homozygous condition which is formed by the continuous selfing. That is about the true breed. Thank you. For more updates on biology, like, share and